I'm Mallory Allison, Editor-in-Chief of Cambridge Health Tech Institute's Pharma Week and Biomarker Breakthroughs e-newsletters. The topic of our webcast is stem cell therapy, and my guest is Bruce Wentworth, Senior Director of Cardiovascular Research at Genzyme. Bruce, thank you so much for joining us. We greatly appreciate it.
Thank you for having me.
Could you tell us a little bit about where your research is focused?
So we have spent a great deal of time in the recent past looking at the capacity of cells to be transplanted into tissues such as the heart. We focused attention on things such as skeletal myoblasts, but also are taking a look at the comparative ability of cells such as mesenchymal stromal cells or mesenchymal stem cells also to engraft in the heart. And ultimately, we hope to learn whether they impart value.
Right, and these are adult cells?
They're all adult-derived tissues. They, in their original concept, would be autologous, but conceivably could be allogeneic, particularly in the case of MSCs.
And why use these cells in cardiovascular research? What's the unmet need there?
Well, in large part, they serve as prototypes. Specifically, in the case of skeletal myoblasts, what we're interested in is, can you add muscle to cardiac muscle? Admittedly, it is not cardiac muscle, but can you add muscle that we know could contract, and will it actually contribute to cardiac output?
Right. So what have we learned so far? What are the most important things about doing this type of therapy in the heart?
There's some very interesting things that we've learned. One of the subjects that we've learned is that cells injected into the heart actually rapidly leave the heart, and it doesn't really matter which type of cell you inject. A large number of them leave right away. We have also learned that injecting cells into the heart, based on the clinical data so far, appears to be relatively safe. I always put that in quotes. Right. It's as defined within the studies that are available, appears to be relatively safe. But we also know that the value imparted to heart failure patients appears to be modest.
Are there a lot of other things changing in our understanding of the biology of the heart? And does this, I mean, right now, are we looking at a new chapter, or is this just a new offshoot of a very standard? No,
there's a whole new realm of data that has been pioneered by folks like Michael Schwartz and Piero Anversa and Eduardo Marban and others who have been investigating the relative importance of endogenous cardiac progenitor cells within the heart. And it's not clear how one can employ those therapeutically. It is only a few years old. It frequently takes much longer than that to just understand the basic biology. Mm-hmm. But certainly it is attractive to think that if one could encourage those cells to grow and replenish cardiac tissue, that would be an attractive avenue for potential therapy.
And what is the scale of the problem in terms of the patients, the type of patients, and what type of problems they have, and how many perhaps?
Yes, I mean, if you go to the American Heart website, you can get a whole compendium of annually updated facts and figures, but it's a $50-odd billion problem in the United States. So it's a big dollar-wise problem with respect to heart failure and heart disease. And there has been a great deal of progress, fortunately, in the understanding of the importance of early revascularization of cardiac tissue to prevent subsequent myocyte death. Yet there still is a great need for those patients who don't get into the emergency room quickly, and many heart attacks are disguise themselves for many individuals. Women in particular mm-hmm. are at risk here. So there is still a great need, because once muscle has been killed, it's replaced by scar, and by and large it's lost. And if the loss is great, heart failure is all too frequently an outcome.
Now, when we look at literature about these types of studies, there seem to be a lot of sort of one-time discoveries where we see, people have had a good response, and that's not always followed up. What's your perspective on this, and do you agree that there's a greater need for better standards within the field for things to become reproducible?
Well, I think there certainly is a better need for standards, and certainly skill in terms of execution of animal studies, and I know many of these animal studies are difficult, and the skill, however, that's needed there is substantial. There's also a need for standardization as it applies to cells, standardization of culture conditions and characterization conditions. And the International Society of Cell Therapy has gone a long way by issuing platform statements with respect to MSCs to guide researchers as to just what they're calling MSCs and how they should be characterized. And that, no doubt, needs to be applied across the board to many cell types, even those which are not concluded to be MSCs or anything else, but the principles therein need to be applied.
Do you think there's some general reason that you see success in animal models sometimes and not then able, I think we were talking earlier about what needs to be in place before people go into phase 1s?
I can tell you what I worry about, and I worry about it with my own studies, and that is the tyranny of small numbers. Specifically, if you do a power analysis, just as you would for a clinical trial, but now your clinical trial is a rat, what you all too often learn as you look at the variability in your test system is that you may well need 20 or 30 animals per group. And all too often what we see are 8, 10, and 15 animals per group.
Right.
It's very easy, all too easy, to actually get a favorable outcome, which is actually not the truth. It's just the way you want it by chance, and you stop. You got the answer, and
Right.
you stop.
Right. So one of the other new ideas that has been discussed is this peregrine hypothesis. What's your perspective on that?
I think it's a revolutionary way of thinking about how cells or anything else might impart its value in the setting of cardiac injury and ischemia. And I think it's clear from some landmark papers that I mentioned earlier, the work of Grunwald et al. in Cell, and there's other papers by Sahin Rafi, which fundamentally outline the factors and the cells that deliver factors into ischemic tissue or injured tissue. And there's no doubt that the peregrine effect must be taking place. The question is, can you actually use this therapeutically? And that's unclear.
Right, and you mentioned that in a lot of the studies you've seen that it's being offered as a hypothesis or an answer to why the study
didn't give the answer desired. Yeah, then that's something one has to really be careful with. If you get an answer that is favorable but you can't account for it, meaning the cells didn't become part of the structure, and you're left saying, well, it's the peregrine effect, you really need to go back to the drawing board and redesign your studies to purposely examine that type of effect.
What is the thing that has drawn you to this field?
Well, that's relatively easy, and I think it's not doesn't just draw me, it draws everyone at Genzyme and probably other biotech and clinical folks that work in clinical settings, and that is that there are patients in great need. And in Genzyme in particular, we spend much of our time serving patients that are desperately sick and with one disease or another. And once you have met these folks, it's not hard to say, well, we ought to go back and try a little harder, see if we can't think of something.
Great. Well, thank you so much for taking the time
You're
to
welcome.
join us. And thank you. These speakers who are joining us for these webcasts are all presenters at our regularly scheduled conferences. For more information about these meetings, please go to healthtech.com. Thank you again.